It's fascinating to me that you could study fruit flies and learn anything about human depression. I'm really dying to know what it is that you observe with fruit flies. Because to me, I'm like, no way a fruit fly has a mood, but maybe they do.
Oh yeah, like there have been several studies with fruit flies, and even some of the very, very early studies with serotonin were in lobsters. Back in the 1950s and 60s, they have a lot of the same neural architecture. And then I have a colleague who studies crawfish, crayfish, crawdads.
Crawfish,
neural effects. And there's a lot of conserved biology for aggression, social interactions. I think that there are going to be populations where the subjective trip is going to be necessary, and those where maybe not necessary. And I think most of them it's going to be necessary. Because I think if you look at a lot of the veterans with PTSD, you look at the crack users, you look at people with major depression and other comorbid disorders, there's a lot of trauma associated with that. A lot of childhood trauma or trauma as an adult that has produced this dysregulation of their HPA axis, and the SSRIs and whatnot aren't really very effective. So the therapies like MDMA, like psilocybin, the subjective experience allows them to really confront, face, absorb that trauma, that history, see it from a different angle. That then subjective trip is over, the integrative therapy is crucial. MK Ultra back in the day were just taking people in off the street and dosing them up.
This was the CIA experiments with LSD. Yeah,
secret.
Yeah, and what were they doing it for?
They were trying to develop psychedelics as a mind control agent, and one of the main leaders of that was at Tulane University, and he was the chair of psychiatry at Tulane. And apparently, they still have one of the bunkers they used to dose people up back in the 50s in City Park here.
Tulane and City Park, I believe it.
And I was several years ago, this person who shall remain unnamed, but he was in his 90s at that point, was involved in these early studies with LSD back in the early 1950s, would talk about how they would take Amish people who would come into the big city and just bring them in off the street, put them into a dark room with no lights, and dose them up with like 500 micrograms of acid and just watch them.
What? That's horrible. That shouldn't be laughing. But the fact that they're Amish, is a particularly weird detail. Like these are the things when I heard these things in high school and in college, I mean, I would be like, that's preposterous. And like, it was like, can you break somebody and get them to believe things that aren't true? And this whole MK Ultra program, that makes sense. So you get someone who's a true believer and see, can you really change their beliefs? Okay, trying this again. Dr. Charles Nichols, thanks for joining me.
Oh, my pleasure. Thank you for the invitation.
So when you meet people out in the world and they ask you, so Charles, what do you do? What's your answer for them?
It really depends on their background, whether they're a scientist or not, or potentially even their political persuasion. So my standard first answer is I'm a pharmacologist. I study medications to treat psychiatric diseases like depression. And then if they have a follow-up, like, well, what kind of medications? So, well, I study psychedelics. And at that point, I'll get, oh, that's really interesting, tell me more. And or a lot of times people will just instantly start to try to tell me the stories of their personal experiences from back in the day.
Trip reports, right? We got to give you trip reports.
Like, let me tell you about that one time I was in college.
Yeah.
So,
so I assume you're saying this is what you get from lay people who are not scientists. When have you seen over the years? How long have you been studying psychedelics?
I started studying psychedelics professionally back in 1997. So.
Yeah, so
it's 30 years.
Yeah. Have you seen a change in how people respond when you tell them what you study?
Oh, definitely.
How so?
I first started studying them back in the late 90s. And I would tell people, and they would give me a skeptical look. Like, aren't those dangerous? What are you studying them for? And at that point, it was, I was studying them to sort of understand how psychedelics work, how consciousness, like the biology of consciousness. And this was really before the therapeutic potential had become widely known. It was about another 10 to 15 years after that.
Yeah, so it was just pure scientific discovery at that point.
Yeah.
What about when scientists ask you, how would your answer be different?
If scientists, a lot of times, I get a lot of the same responses. If I tell a scientist, they'll go, "Oh, let me tell you back in the day in the 60s when I was in graduate school," or something like that. But with scientists, it gets more a bit mechanistic. Like, "Oh, wow. So tell me more. How do they work? What are the neural circuitries, receptors?" So it gets a bit more detailed. But across the board, people are much more interested in actually, follow-up questions. How's it working? What's it doing? So it's been going from kind of a skeptical, "You're doing what?" to, "Oh, that's so cool. Let me tell you about my trip," or something like that.
Yeah, I mean, have you seen? Was there a certain turning point at which point the scientific research into these medicines became more, I guess I'd say, socially acceptable, like more professionally acceptable? Because I think for a while there were a lot of people in science and in academia that wouldn't publicly admit that they were even interested in them.
Oh, yeah. I think it was about the mid-2010s when I was a postdoc and when I was first trying to get grants as a new assistant professor here back in the early 2000s. We were studying psychedelics as tools to look at the target 5-HT2A receptor and potentially how it was involved in schizophrenia and psychosis. So we kind of had to we want to know how psychedelics work and understand consciousness, but we had to wrap it in this context of using them as tools to understand what goes wrong with consciousness, like in psychosis or schizophrenia. And those studies were very helpful and informative on understanding kind of the molecular basis of psychiatric diseases. So yeah, don't get me wrong, they were very valuable. But most of us in the field at that time, there were maybe, 6 or 7 of us, we were all kind of taking the same approach because we couldn't study psychedelics to study psychedelics.
Right, just to understand the nature of consciousness. And also I imagine that your funding sources, I imagine, are mostly from like public sources?
Yeah, at that time, the NIH, primarily the National Institute of Mental Health, they were funding a lot of different investigators to study 5-HT2A receptor pharmacology. Like the lab I did my postdoc in, and several others, but it was really kind of fundamental neuroscience. And how does it relate to psychiatric disease like depression or depression, substance use disorder, schizophrenia, things that serotonin were involved in. The serotonin 2 was highly implicated. And so we were studying potentially its dysfunction in these disorders and using psychedelics as a tool.
So you've said these letters a few times, 5-HT2A. Can you tell us about what the significance of those numbers and letters mean?
So 5-HT is the chemical abbreviation for the neurotransmitter serotonin. And serotonin was first discovered and isolated from something like 5,000 liters of cow blood back in the 1930s as a molecule that was able to cause vascular tone. So they took the serum, they identified this factor, they didn't know what it was yet, but when they put it on, say, an isolated blood vessel, it caused it to constrict. So they named it serotonin. And a few years later, they had determined what the structure was. And the structure of serotonin is 5-hydroxytryptamine. So the tryptamine core with the 5-hydroxy, so 5-HT. And that defines the neurotransmitter. And for serotonin, in mammals, there are 14 different families of serotonin receptors, each encoded by different genes. And then there are different splice isoforms and subtypes and edited isoforms of those. There's all of them are what are called G protein coupled receptors, with the exception of one of them. So that means that the receptors sit on the surface of the cell, and when serotonin binds to it, they transduce a signal to the inside of the cell that then activates all kinds of enzymes on the inside of the cell to change its physiology. And there's the main families are serotonin 1, 2, 3, 4, 5, 6, and 7. And the primary target for psychedelics are serotonin 2 receptors. And in this 2 family, there's serotonin 2, the A, the B, and the C. And studies in animals and humans have shown that it's the serotonin 2A receptor, the 5-HT2A receptor, that is the critical one for the psychedelic behaviors of these drugs. If you block them in humans with a selective drug that prevents that psychedelic from binding to that receptor, you don't get any of the behavioral effects.
So are there any other like medications that are used that would attach to the same receptor?
Oh, yeah. So that's one of the reasons we studied the 5-HT2A receptor and psychedelics to study schizophrenia. Is because one of the primary classes of antipsychotic medications called atypical antipsychotics that were developed first in the 1980s have as a major component blockade of 5-HT2A receptors. So the older antipsychotic medications primarily just block dopamine receptors, and they would stop a lot of the positive effects, the hallucinations, and then the next generation of antipsychotics that came along had less dopamine, but then more activity to block serotonin 2A. And I think more people are taking those than the classical. They're safer. And there's even a newer drug that's come on the market over the last couple of years called Nuplazid, which is a selective 5-HT2A blocker. It doesn't block anything else. And that's used to treat psychosis associated with Parkinson's disease.
Interesting.
So you've got evidence on one hand, if you activate the receptors, you produce these profound reality distortions, hallucinations. But if you block that receptor, you can prevent the same types of...
That's fascinating. I mean, yeah, so it's almost like I think of it like on my iPhone when I do photography, there's a slider for contrast, black point, vibrant, saturation, and you can just kind of dial up an effect or dial it down. And the slider appears to be this receptor, which is kind of amazing. And also with Parkinson's, I mean, my first thought with Parkinson's is dopamine. You would think that would be the mechanism of action. And it's interesting that serotonin also seems to be involved. And with psychosis as well, I didn't know that.
Right. It's involved, I think, directly and indirectly, because one of the things about the serotonin 2 receptors is that they will modulate dopamine tone in the brain. So 5-HT2 activation leads to increased dopamine. 5-HT2C activation leads to decreased dopamine in the brain through indirect pathways. So it's a really complex pharmacology. Yeah.
So do you think that, like, as a general matter, that dopamine and serotonin kind of work antagonistically, or is it just at these specific receptors that they seem to have that effect?
Oh, they can act synergistically, antagonistically, depending upon where they are, location in the body, the nature of the receptor. There are stimulatory and inhibitory dopamine receptors. There's stimulatory and inhibitory serotonin receptors.
It's endlessly complex, right? Do you think we'll ever fully understand all of it? So we had some technical difficulties. What we were talking about was, you were just saying that, there are inhibitory and stimulatory, versions of dopamine and serotonin. They work together synergistically and antagonistically. And my question was, do you think that we'll ever fully understand the neurobiology of the brain?
Do I think we'll ever fully understand it? No.
Yeah. Do you think there's just a theoretical limitation on our ability to do that?
It's extremely complicated, and I think, even if quantum AI comes online, I think the brain is still an orders of magnitude more complicated. I think we can model circuits, maybe at some point come up with artificial intelligence that's super intelligent, but in terms of understanding the complexity of the brain and everything that's there, I don't think so. It's like the more we know about the brain, the more we know we don't know about the brain.
Yeah, because I mean, it's like exponential complexity. So everything new that we discover comes with an exponential amount of more complexity that we don't understand, right? Yeah. So it's just, I mean, and I'm just still, I've been kind of ideating about it for years, just trying to figure out the basics. I guess it's nice to be able to talk to you because you can explain, the term neurotransmitter. What exactly does that mean?
So a neurotransmitter is a chemical hormone, or it could even be a peptide, that's released from a particular type of cell or tissue in the brain that transfers information from one cell type to another cell type by a receptor. So in the case of, say, serotonin or dopamine, those are presynaptic, postsynaptic. There's, like a releaser and an acceptor. And when the, say, presynaptic cell receives a signal, an electrical signal to fire, it releases its neurotransmitter to the other side. And there are receptors on the other side that capture those neurotransmitters and then reinitiate signaling on the backside.
A new electrical signal.
It can be. So in the simplest, you would get an electrical signal would come to the presynapse. That would open voltage-gated calcium channels. Calcium would come in. The calcium that comes into the presynapse would then, through a series of reactions, cause these neurotransmitter vesicles to fuse with the presynapse. They release the neurotransmitter. If it's an excitatory neurotransmitter, say it would bind to the postsynaptic neurotransmitter receptors that would then increase calcium. That increase in calcium then would continue to propagate that action potential down to the next neuron. So that's sort of simplest. But what you have is on the presynapse, you can have multiple neurotransmitters released. They can be excitatory, they can be inhibitory. You also have negative feedback on the same presynapse, so that when you get a lot of neurotransmitter released, you get diffusion around to the extrasynaptic area, which can then cause negative inhibition of release. And for the serotonin system, you have the excitatory or stimulatory serotonin receptors on the postsynapse, but on the presynapse, you have serotonin 1B and 1A receptors, which are inhibitory. So when you get serotonin is releasing, when you've got a lot of serotonin in there, you've got this sort of auto feedback negative regulation to then damp it back down again. So you just don't get runaway neurotransmission. And there are...
So it's almost like feedback in a microphone. Like if you have a microphone and an amp, there's like an automatic cutoff when it knows it's escalating. It's like, we're cutting that off.
Yes, exactly.
Basically, which is appreciated when people have that.
Right.
And so it's just, that's amazing. It makes me wonder, at what point in the evolution of life did that feature come along? I mean, one of the things that I've found very interesting about your work is that your work in serotonin is not limited to the human species.
Oh, no.
Yeah. Tell us about that.
So my PhD dissertation research was in fruit flies. And at the time I went to graduate school, it was in the late 1980s. And PCR machine, gene expression, it was kind of real early days of truly understanding what genes were and elucidating how interactions between expression between genes would regulate development and brain function. So I was in the laboratory at Carnegie Mellon University, and we were studying the retina development of the eye. And the primary mechanisms involved in the retina development of the eye actually were instrumental into understanding how a lot of cancers in humans progress. And so studying the fly sheds a lot of information to human biology, but not necessarily like you would think. Like, who would think studying the fly eye would lead to developments to treat cancer? I'm sure the people who started studying it probably didn't go looking for that either. That's one of the reasons why people kind of lack an understanding of why we do basic research. Mm-hmm. Because you find out about stuff like that. I mean, you study the eye of a fly. I don't know. Maybe did people go looking for cancer? Like, I'm sure there's a growth factor or something, right? No. It was my dissertation advisor was John Pollack, who had done his postdoctoral work for Seymour Benzer at Caltech. And they were looking at what the signals were in the fly retina that caused this undifferentiated cellular epithelium to turn into neurons and support cells. And they elucidated this pathway called the RAS pathway that was underlying how a particular type of photoreceptor in the fly eye developed from just this undifferentiated cell to turn into a photoreceptor cell. And it required this complex signal transduction pathway of like 15 different enzymes to happen. But as it turns out, the receptor on the undifferentiated cell in the fly is the same kind of receptor in humans that mediates a lot of cell growth. And the entire pathway from that fly eye receptor receiving the signal to go tell that cell to change its developmental pathway is the RAS pathway, which was identified in many human cancers. And so the entire pathway of the human cancer RAS pathway was first found in the fly by not trying to study cancer, but through just basic biology. And that's
just
how
understanding
discoveries are made. It's
basic science.
Yeah.
And so you can do that in flies. And so so from your doctoral research, I guess you built an expertise in fruit flies. Is that the specific species? And then so how did you go from there to psychedelics? I mean, I can imagine, but I'd really love to hear the real story.
So I was really enjoying fruit flies. I learned a lot about genetics, about developmental neurobiology, development in general, really basic science, but I was not enjoying taking care of all the flies because you can't just freeze flies away, thaw them out, and use them again. And I really was just working with RNA and DNA, and I wanted to do something behavioral, more with mammals. And so I started looking around for a postdoctoral position in some kind of mammalian behavioral biology to get away from flies. And I had looked at a few different opportunities, and just through a series of serendipitous events, ended up in a lab at Vanderbilt. Elaine Sanders Bush, who had been studying psychedelics since the 1960s. And she was very instrumental in understanding how the target receptor for psychedelics, the serotonin 2A receptor and 2C receptor, fundamentally function and the signaling pathways that underlie them. And the first project when I joined her lab was to investigate gene expression changes induced in the brain by LSD in rats. And I thought that was really exciting because it really combined my knowledge of developmental neurobiology and gene expression with something that I personally thought was an interesting topic, like psychedelics. But I had never intended to get into the field of psychedelics. That was never in the cards. I was really interested in molecular biology, genetics, and this opportunity just arose.
Now, your father also is a psychedelic researcher, right?
Yes. Yeah.
Yeah. And so were you, I mean, I'm sure you were aware of what he did at some point, but maybe wanted to differentiate yourself professionally?
Yeah. The whole time growing up, I knew he was a chemist, a medicinal chemist. Although I wasn't quite sure exactly what he did chemistry on. When I was younger, and like, what kind of chemicals do you do? He would say, I work on chemicals that make people dream. I was like, okay. And that was sufficient for somebody who was like 7 or 8 or 10. And it wasn't really until I was probably about a junior in high school when this was probably around 82, 83, when he was telling me, okay, these are the types of chemicals that I work on. And this was back in the early Reagan era. This is your brain on drugs. So it was more, I never did get any of that negative propaganda. But it was like, oh, my father works on these chemicals that hippies used back in the 60s. Okay.
Yeah.
Yeah. So I thought it was
so it's kind of available when when you find this lab here at Vanderbilt.
Yeah. And when I did my college undergraduate, I started off as a chemistry major because I really like chemistry. And then when all the molecular biology hit and Purdue got their first PCR machine, that became more attractive to me. So I switched my primary major from chemistry to biology. But I continued on the chemistry and kind of got a double major in chemistry and biology. But then when I graduated, I was really enamored with genetics and molecular biology, so I went off to do that for graduate school.
There's a few terms here I've been kind of racking up in my head that we need to define, because I've heard them and I've never really gotten a good definition. Why don't we start with molecular biology?
So that's kind of changed in definition over the years. Molecular biology, I would say, really is to the biology of molecules within a cell, enzymes, like DNA splicing, how to make transgenic cells or animals, when you're doing biology to understand how cellular components work. Not like biochemistry and molecular biology have a lot of overlap. Biochemistry is looking at kind of the chemistry of how these enzymes work. Whereas the molecular biology can more encompass like the process of DNA replication, of cell metabolism, but it really focuses on the biology of processes that happen within a cell.
Okay. And then, so the next one also is gene expression. I've heard that term a lot,
right.
But I'd love to hear your explanation of what that means.
So gene expression has a very long history. Like back until like the 1930s and 40s, we knew that heritable material was carried on chromosomes, but we didn't know how that material was transmitted. We didn't know whether it was on the protein that was in the chromosomes, whether it was on the DNA that was in the chromosomes. And somewhere along the line, the term gene was coined for the heritable unit on a chromosome, the piece of a chromosome that transmits information from one generation to the next. And then as we became to understand that genes were encoded by DNA, then people started looking at the structure of the DNA, how the structure of the coding versus non-coding regions on the DNA contributed to the proteins that were made. So when I'm referring to gene expression, it's like, how are the proteins that are made in the cell that are relevant to the cellular function, how are they encoded in the DNA, and how are they turned from kind of like beads on a string and spliced together to form an active protein unit?
Yeah, it's interesting. I just got the visual metaphor of like a piano. Like there's chords that you can play.
Yeah.
And different chords are going to produce different sounds. Like there's a number, and there's not an infinite number of possibilities, but there's a lot.
Right.
And if you consider all the keys on a piano that you could play, and like you could select just a certain assortment of genes, and they're going to produce a very specific protein for a specific purpose. And when I've always heard gene expression, I've heard in the context, diet is a common one where I've heard like you can, there's a lot of talk about ketogenic diets will cause different gene expression. To me, like I think growing up in the 90s and learning about genetics at that point in middle school, you think that like you have your genes, you got them from your parents, and then that's your genes. And we don't really think about them in terms of being activated or expressing or not expressing. But that's basically, they're always in communication with our environment, right?
Oh, yeah. And it's very, very dynamic. So it used to be that, there was one gene, it was expressed, it made a protein. And then we come to understand that a gene wasn't a single unit. There were introns and exons. That a gene was, the terminology is a locus, that the pieces of DNA that together can be turned into protein, a functional protein, are tiny pieces of DNA along the locus that some of them are turned into protein and some of them aren't. And in fact, the gene that I worked on primarily when I was a graduate student was called the lozenge gene, had such complex genetics in splice patterns that it was one of the main genes that was used by Ed Lewis in his formulation for the hypothesis of introns and exons and gene structure. And so my dissertation research helped to sort of clarify what these advanced genetics are and how you can get a gene locus expressing from the same piece of DNA one variant of a protein in one tissue, but another variant of a protein in a different tissue that has a different function. And so
from the same gene.
Right. So you can string together different components to get a protein that can have different functions. And that seems to be common with about most genes and proteins.
So this is the kind of the science that you were doing when you got here to Vanderbilt and were working on your postdoc was on gene expression with LSD. What can you tell us about that?
Right. So what we were trying to do is look to see in the rat prefrontal cortex, which we knew that's kind of where all the magic was happening with the behavior of LSD due to other experiments from other laboratories. What kind of effects was a psychedelic like LSD having on what genes were being turned on and what genes were being turned off? And by knowing what the response was at the genes that were being turned on and turned off to LSD, we could kind of deconstruct that to get a better idea of what LSD was doing to a cell. Because it was completely really unknown. We knew that LSD was interacting with specific 5-HT2 receptors, 5-HT1 receptors, that it was turning on certain signaling pathways, but beyond that, what effects were it having? Was it inducing, say, genes that would indicate that there was cell damage going on, or plasticity? Nobody really had any idea. So it was a way of further investigating and drilling down to a deeper level of understanding of how LSD affected the cells in the brain beyond just the receptor interactions and acute effects.
And what kind of discoveries did you make at that time, or were made at that time when you were doing that research?
So we didn't really know, given all the propaganda that had been going on with "this is your brain on drugs." So one of the findings we might have expected were we were getting all kinds of toxic markers showing up, like a cell. There are certain genes and proteins that are expressed when a cell is damaged. And if those are expressed, you can say, oh, this drug it's toxic. It's damaging the cell. It's damaging the neuron. So that was one possibility. And we gave these rats an incredibly high dose of LSD just to make sure that we were going to see whatever we saw. So this would be like a person maybe taking 10 to 20 hits of a recreational dose of LSD, and then take
which is just so like LSD is dosed by the microgram, right? Like that, it's incredibly strong. So you're saying a recreational dose, maybe 100 micrograms?
So a recreational dose, say 100 micrograms. So the equivalent of what we gave the rats would be like 1,000 micrograms.
That's a lot. Yeah. So there's no doubt that they've taken.
Oh yeah.
LSD, right.
And you look at them; they just look stoned. There's a stereotype behavior in a rat that you can tell they've
interesting
psychedelic.
And that's part of what you were doing here is moving into the behavioral biology, right? So you're literally observing behavior, which I kind of tend to agree is much more interesting.
Yeah.
To see like what did you observe with rats when you said they look stoned? Did they just kind of lay on their back and like soak up the sun, or
almost? There's a set of behaviors. One is called ptosis, which is your eyes are kind of like half closed. And the other is called hind limb abduction, which means they can't hold themselves up. And they kind of look like they're swimming a bit. And it's very, very classic of a rat on a psychedelic. It hangs out, its eyes look stoned, and its body posture is really kind of, it looks like it's sacked out on a beanbag.
So
it's not.
My first question is, do you think they find it enjoyable? I mean, if to the degree that question makes sense with a rat, which I think is the bigger question here, is what is a rat consciousness really like?
To an extent. So they don't really exhibit signs of overt stress. While they're under these states, they'll play with their cage mates, they'll eat they'll groom. They don't really exhibit classic signs of a stressed animal. And these behaviors are transient. They go away after about half an hour.
Interesting.
So we took the brain
with LSD on rats. That seems to be the primary effects are a half an hour?
Yeah. You can measure the half-life of the effects of LSD in a rat are about 90 minutes. So if you think about it, rats evolved living in refuse and garbage and trash, and so they're exposed to a lot of toxins. So they have a very high metabolic capability compared to humans. So,
and that's probably
rats and mice
drugs in general. They're
just
fast.
Chew through them really fast.
What was I about to ask?
So what we found,
you mentioned toxicity also. I was just wondering about that as well.
So we first tried this technology, which turned out to be a bust, and then microarrays were invented. So we did some of the very first microarray studies, and with those we're able to look at like 10,000 different genes on a gene chip and look for differential expression. And what we found was really interesting. Despite given this rather high dose of LSD, the genes that were coming back as differentially expressed were primarily all increased in expression. We didn't find any that were decreased. And they were all genes involved in synaptic plasticity, like learning and memory. Genes that would be associated with when you're consolidating a memory or when you're learning something. So that was a first clue that psychedelics might be helpful for something like the process of synaptic plasticity and learning and memory. And that was kind of the first evidence of that. And we published on that back in 2002. And if you fast forward to where we are now, LSD and these others are now psychoplastogens that enhance synaptic plasticity and learning and memory. We first had clues of that back 25 years ago from these studies we did at Vanderbilt.
So what is the word synaptic plastogen? I think synapse is the thing you've already explained. It's kind of the gap, between the space between things, literally. What is a plastogen? What is plasticity?
So when you have a neuron, it's imagine like a Christmas tree that's decorated with branches, and on the branches, the ornaments, those would be like the synapses that are connecting with other neurons. And this is all in three-dimensional space. When you're stressed or under certain kinds of conditions, you have a decrease in those connections. Or if you're stimulating that neuron and using it a lot, you have an increase in the connection. So it's very dynamic in the number and the nature of connections you have between the different neurons in a particular circuit. And that process is covered under the umbrella of plasticity. It's the ability of one neuron to change the nature and the number of its connections with another neuron. And those can be measured by a microscope and literally counting the number of connections between two neurons, or a functional measurement using a microelectrode that you can stick into a circuit. And there are certain, if you're getting long-term depression, LTD, or long-term potentiation, LTP, as you're injecting current into a circuit, you can sort of have a forward feedback or negative feedback and look at the function of it, which is reflective of what's happening at the cellular level. So when I'm referring to plasticity, I'm referring to the ability of a drug to enhance the functional connectivity between neurons.
It's the ability for the neurons to connect, and we kind of need chains of neuronal connection, right? Neural pathways to do things, to swing a golf club, to think, to be, to remember.
Like when you're learning,
it's also
you're enhancing the connectivity in those circuits underlying that behavior.
And so go ahead.
Oh, and in depressed individuals, stressed individuals, PTSD, major depression, they've shown that the connectivity between certain circuits in the brain is less than a healthy, normal individual. So there's a deficit of functional connectivity. And so the recent clinical studies and neuroimaging with psychedelics have shown that, and in the preclinical work that's also been done, that a single administration of a psychedelic like LSD, like psilocybin, can restore the functional connectivity in these deficient brain circuits. And then the hypothesis is that's what's underlying the therapeutic effect.
The restoration of plasticity,
right?
Basically,
right.
So there's also you mentioned drugs can create it, but I've also think about exercise all the time as a modulator for plasticity. And there's this neuroscientist at NYU. She's the dean of the Arts and Science College now, Wendy Suzuki, and she studies the neuroscience of exercise. And she calls exercise a bubble bath for the brain. And I love that because it's probably highly regulating, literally, of all the neurotransmitters. Because I think it's probably the main thing we evolved to do is move. We're distance runners basically, and so it's interesting. I always like to exercise and then meditate immediately after. So my brain's the most plastic for that little period after. But it's interesting. I mean, you're working with flies, or you're working with rats. You find that there's these things about learning and memory, which I imagine must have been really surprising to associate that with LSD in the early 2000s. But then, when I'm thinking of psychedelics and also in serotonin, I think if the average person, if you did a word association between serotonin, if they provided they understood the word, they're probably going to say something like mood or depression. And it's interesting that we're getting learning and memory, and it's like, okay, well, what are the relationship to that to mood?
Yeah, it's complex. Serotonin regulates learning and memory. It regulates mood. And some of the work that we actually did in fruit flies several years ago here in New Orleans was looking at the role of psychedelics and different serotonin receptors in the process of learning and memory in the fruit flies, and showed that there's a very important role that is similar to the role of serotonin in humans for learning and memory. So what
can you tell us about that?
Absurd biology, like the serotonin 1 receptors, serotonin 2 receptors are both intricately involved in learning and memory in both humans and mammalian systems, and also social interactions. And with respect to mood, we published a paper last year on the antidepressant-like effects of psilocybin in fruit flies. So whatever these mechanisms are that are producing long-lasting effects on, say, mood and learning and memory are conserved very, very far back in evolution.
Yeah, so I'm dying to know about that because, I read this paper about some years ago by Charles Nichols called "What Is It Like to Be a Bat?" Are you familiar with this?
No.
It's kind of, I think, if it didn't begin the field of philosophy of mind, it changed it profoundly, because it's this essay about, can you really imagine what it's like to be a bat? But he kind of essentially posits this definition of consciousness, which is that for something to be conscious, it must be something to be like that thing. Like there must be something what it is to be like a bat. And we don't really know what that is because we're not a bat. Like we don't have the neural architecture to be able to understand it. But a bat has an experience that is very bat-like, and it's just kind of a profound. It's a really short little essay and paper, and it's like really mind-numbing. But it does make me wonder a lot about studying things like consciousness. It's fascinating to me that you could study fruit flies and learn anything about human depression. And it's kind of wonderful and amazing that you can do that. I'm really dying to know what it is that you observe with fruit flies. Because to me, I'm like, no way a fruit fly has a mood, but maybe they do.
Oh yeah, there have been several studies with fruit flies, and even some of the very, very early studies with serotonin were in lobsters back in the 1950s and 60s. They have a lot of the same neural architecture. And then I have a colleague who studies crawfish, or crayfish, crawdads, looking at
crawfish.
Neural effects.
You're in New Orleans, right?
Crawfish. Yeah. Looking at the neural effects of those, and there's a lot of conserved biology for, say, aggression, social interactions. Like, for example, in the crawfish, if you've ever been to a crawfish boil and the crawfish falls out of the pot, you go, oh, that's not good. I want to eat you. You want to put it back in the pot. You reach down to get it. Its claws are going to go up. It's going to pinch and it's going to back away from you while you're trying to pick it up. That behavior is mediated by activation of serotonin 2 receptors in the crawfish. So the same receptor that is the psychedelic receptor is sort of that aggressive stance.
So if you gave a crawfish high dose of LSD, do you assume that's what it would do?
Oh, it might. It might have been done. I have to look at that.
I'd be curious. Or would it just do it at everything, and regardless of whether someone was coming? Yeah. I mean,
a lot of the serotonin work is really translational. There are several labs that have looked at aggression.
Translational means in other species, and then
right, what you find in a human, you find in a model organism, you also find the same fundamental mechanisms in a human. Like
for ethical reasons, it's easier, I assume, to do these things to rats and crawfish than it is to people.
Right, and people think of serotonin as the happy drug or the happy neurotransmitter, but it's the homeostasis. It's a set point. When you've got too little serotonin, it might produce mood disorders, depression, anhedonia. If you've got too much serotonin, that's linked to hyperaggression, aggressive outbursts. So it's really not about having the neurotransmitter there or not. It's about how much you have. And how much you have can produce opposite effects on behavior.
So one of the things I'm very interested in and have been for a while is just like with psychopharmacology are psychotropic drugs for mood and things like that, especially antidepressants. We have a lot of antidepressant medications in this country. And going back, I think at the beginning of the 70s, there's been this idea of the chemical imbalance theory, which most people you talk to in medicine, will kind of privately admit that we know that that's not how that works, really. But there's just the way you just described it actually does seem like balance really is the relevant factor. And it's not like that. I think most people think, well, your serotonin's low. You take this pill, you get some more serotonin and you feel better. And I don't know. Is that really how it works, do you think? Or obviously it's more complicated than that. But I'm just curious.
It's more complicated than that. And the serotonin deficit theory is still controversial. There have been some papers that have come out recently saying that that's not actually what happens. But then you look at the methodology and it wasn't quite right. There are those that are. And it's not so much like if you take an SSRI like Prozac or citalopram, its primary mechanism of action is you're blocking this protein on the presynapse that once serotonin is released, it's responsible for taking this free serotonin that's not bound to a postsynaptic receptor or not metabolized away to bring it back up into that presynapse and repackage it like it's a recycling. And what the SSRIs do is they block that and increase the levels of serotonin in the synapse, making the concentration that's exposed to the postsynaptic neurotransmitters higher. But that's not all these SSRIs do. They also have affinity for other transporters. They have direct affinity for certain receptors, like 5-HT1A. So they're affecting levels of serotonin, of norepinephrine, of dopamine, and they're directly activating like serotonin 1A receptors. So it's not just that these SSRIs are enhancing serotonin and that's what's fixing things. That's a part of it, but they're all doing many other things. And when it comes down to it, we have absolutely no clue what the primary mechanism of action of an SSRI is. And the FDA states that you don't have to know how a drug works. You just have to show us that it works and it's safe. And so there are so many drugs on the market that we have no clue how they work, but they're safe and they do work.
Right. Yeah. So the way what you were just describing is like reuptake inhibition. I think a lot of like mood and mind altering drugs seem to work that way. Yeah. Is that a fair statement? I mean, I think even like that's how amphetamines basically work with dopamine and norepinephrine. And it's always interesting. I mean, something's always about inhibiting the inhibitor. Or like it seems in medicine, like you find a way to block the thing that inhibits something, and so you get more of it, or it's like these little indirect chains. Kind of, I envision like a Rube Goldberg machine. You know how those work, where it's like the thing. It's like a cascade of things. And going back to that piano analogy with the genes, it's like if you change one note in a piano chord, you get a totally different sound,
right,
and a different mood effect. So it's amazing. It's almost amazing that we know anything.
Yeah,
really.
I'd say that's a really good analogy. Like the brain is the largest Rube Goldberg machine that you could think and ever would exist.
To trillions of connections.
The piano, it goes down to the level of if you have a receptor and you have a molecule, and you change one atom on that molecule, it still activates the receptor, but now it activates that receptor differently. So instead of turning on pathway A, it turns on pathway B, and maybe turns off pathway C halfway. So even at the level of adding one methyl to a compound can completely change the output of that giant Rube Goldberg.
Yeah, that what you just said it's kind of chilling, because that's how a lot of quote unquote legal marijuana works. I mean, there's a big problem with that here in Tennessee, where we still have not allowed just natural organic cannabis to be sold. But so it's a lot of like Delta 8 THC, which you can just go buy at some head shop. And there's what I've noticed when I moved here was something that I moved here 4 years ago in 2022. I began to see a lot of something that I thought was an urban legend, which was cannabis psychosis. And I mean, I'd heard of like one event from someone that had taken like Spice or K2, but this is like mainstream. It's a real problem. It seems to predominate in men under 27, but it just was not something I'd ever experienced before in the previous decade of just using lots of real cannabis. And it just seems that people are just, because in our laws, the statutes will ban a chemical compound. So people will just alter the chemistry. They'll add a methyl or take one away or whatever. And it's like, but there's really bad unintended consequences. And this is a terrible way to do drug policy, but it's what we have in the United States.
That's like, there was once, I think it was on PBS, it was a special on the interaction between the plants that shaped human lives. And they talked about corn, wheat, and one of them was cannabis. And they made an interesting point that until cannabis was made illegal and scheduled, it wasn't very potent. It took a lot, you had to smoke.
You could just kind of smoke a joint for a while. Yeah.
When they made it illegal, that it really turned it into like all the people who were experts in genetic engineering and plant engineering and botanics, all focused on increasing the diversity of the cannabis strains and increasing the percentage of cannabis and allowing them to grow under grow-like conditions instead. So they're like the epitome of plant genetic engineering was focused on cannabis. And if it hadn't happened, then we wouldn't have developed these super potent cannabis strains, which are out there now, where they're like 10 times, 20 times more potent than cannabis in the 60s. Like one puff now, as opposed to
could have life-altering effects. Yeah, it could give you a panic attack. Or
the cannabis is so potent and produces a lot of these psychosis reactions, because it's like having same can be seen with caffeine. You've got these monster shot drinks with 600 milligrams of caffeine. You're getting more and more incidents of caffeine psychosis presenting.
Interesting. I did not know that. Caffeine psychosis. God, that sounds horrifying.
And it's the same with like the methcathinones and the bath salts. They make one illegal, then another one pops up. When if they just would have left it alone, we likely would have just had that original 1960s cannabis that wasn't getting anybody very high.
Yeah, well, it's funny because this is like, when you create a massive profit incentive, also like if you look at the flower from cannabis, the bud, which is, it's like a delivery vehicle for a drug, and you're going to charge money by weight, but you also have to move it by weight. It makes more sense to increase the concentration of the drug.
Yeah.
It's just like in that, that's why cocaine is a better drug smuggling product than cannabis, because it's just way more potent per unit of weight. And so you get all the best minds in botanics and everything just working on it like it's the cure for cancer, literally.
Yeah.
And, and so this is what you get. And it is kind of like, I wonder what we would have done if we had directed that human potential towards a better use. But these are kind of the unintended consequences of drug policy that, I've seen ever since law school, of just seeing like, how, and it's a lot of people will say, well, it's like, what, should we just legalize everything and have a free for all? And it's kind of hard to be like, because it's like, no, that doesn't sound necessarily good either. But there's got to be, a better path than when we create these massive profit incentives. It just seems like innovation and science are much, much more nimble and faster than our legislatures and our regulators. You know what I mean? And so we're just playing whack-a-mole. And it does seem like that. You're probably really familiar, way more familiar with this than me, is like the research chemicals. People will buy research chemicals from China or India or wherever they can get them. And a lot of them are like in the psychedelic realm, or maybe not. I'm not really sure. They're altering little things. What can you tell us about what people are doing with that?
Yeah, well, those, I think for a long time, when my father still had his laboratory active, it's like every time that he would get a publication on some molecule that he had developed, it would then show up on the research chemical market. And I think he had somebody ask him once, he should be ashamed of publishing knowledge because it was killing people. And he's like, my job is to figure out how these receptors are working, to understand the brain. It's up to each individual to decide what they want to put in their body. And if they want to take a toxin into their body, that's not my fault. So it has been an issue. And a lot of the research chemicals were either published in TCOL, from my father's laboratory, and then from a few underground chemists who have come up with stuff. But it's a real issue, and these are untested compounds. We don't know what their safety is. For example, the compound 25-CN-NBO was a research chemical that actually my father did not invent. It was somebody in Germany who invented it, and that was making the rounds 10 or 15 years ago as the super potent LSD derivative that was killing people. And we had tested it in my lab, and it was the only 2A agonist that we ever tested in my lab that killed my experimental organisms. So there was a known toxicity with it. And, that's
really something we haven't really talked about. With psychedelics, they generally, like the classic ones, don't have a lethal dose.
The ones that are characterized, the natural ones,
right.
And these chemicals that we're coming up with, they're psychedelic, but who knows what else they're doing? Like this 25-CN-NBO, the toxicity, I did some testing on it, and it was a non-5-HT2A mediated effect. So it's not from the primary. It's some off-target effect is being toxic. And these research chemicals that are out there, nobody knows what else they're doing. And they could very well be producing some toxic byproduct or inducing a toxic effect. So it's really playing Russian roulette with these research chemicals. And I think
it's also a function of prohibition.
Yes,
because they're legal. Like you can just order them on the internet.
Yeah.
I want to go back to asking about the caffeine-induced psychosis. That's fascinating, and also with the cannabis-induced psychosis. Since you've studied psychosis and schizophrenia, I'm just curious. Like, do you have any idea of why just a much larger dose of something that you would ordinarily take, like caffeine or maybe cannabis, but in a much higher dose could trigger psychosis? Do you have any sense of how that's happening mechanistically?
Oh, so it's like a high dose of any kind of sympathomimetic CNS stimulant. Cocaine, amphetamine, caffeine will all produce overstimulation of the brain circuitry. And at this overstimulation, it converges on these psychotic-like effects of paranoia, pounding heart. So at higher doses, people would associate, if you take too much meth, you have meth psychosis. You're going to go into the psychotic break. You take too much amphetamine, you're going to have a psychotic break. And maybe not people enough realize that if you do that with caffeine, it's the same is going to happen. And if somebody presents at the emergency room with caffeine psychosis, they're going to have exactly overlapping symptoms as with somebody who's taken too much, say, amphetamine. They're going to have the same body symptoms. They're going to have the same paranoia, mental symptoms. And it's a lot of times, like, people take a cup of coffee is 100 milligrams. And I just heard there was a report yesterday that came out from a group that looked at the maximum amount of coffee intake a day for health benefits. And I think it was 5 cups of coffee a day. Anything more than that, and it's detrimental to your health. And that's still 500 milligrams. But you have these monster shots, energy shots that have all these neon lettering and marketed to kids, and they've got 500 or 600 milligrams of caffeine in it. And if you have, it's a shot. You take 1, take 2. If you take 3, you're going to be having you could literally become psychotic. And can these effects be permanent? I mean, once you kind of tip into psychosis, now you're kind of a candidate, right? Well, I think you just have to cool down.
Okay.
I don't think they're...
And that's what antipsychotics do too, right? I mean, they're just essentially depress
dopamine
everything.
System. But for the caffeine, if that's what it is, you just have to wait it out. It's not going to really produce any damage. And few people know this, but Roland Griffiths, who his claim to fame later in life was at Johns Hopkins with all the psilocybin studies. He also was one of the world's leading experts on caffeine. And when I give the lecture on CNS stimulants and caffeine to the students here. And when I was putting those lectures together, I noticed that a lot of the earlier caffeine studies were published by Roland Griffiths. And I had a few conversations with him on that. And he said it was an interesting story that he relayed to me, that now how the amount of caffeine has to be listed on any given drink. Like if you look at a can of Coke, a can of Red Bull, it'll tell you how many milligrams of caffeine. Well, Roland was the one who led the charge for that, because he had recognized that you had all these drinks that were being marketed to children with caffeine in it, like 500 milligrams of caffeine, and nobody knew how much caffeine was in it. And so he testified before Congress on adding the amount of caffeine to the packaging. And he said he was getting death threats. He had the major beverage industry were taking out like PR hit jobs on him. It was quite brutal on him for a couple of years. And then finally, after his congressional testimony, it was mandated that you must have the amount of caffeine on these containers for people to know what they were taking.
Yeah. Well, it's one of my favorite trivia questions to ask people is, what's the most commonly used drug in America? And they never think caffeine, because they don't think it's a drug. But it's clearly, it's a stimulant. It kind of depends on what's your definition of a drug. And for most people, what's your definition of a drug is something that only bad people do. Or that's basically because of like this is your brain on drugs. That if something's illegal, it must be bad for you, and if it's if it's not legal, it must be good for you. But we know the things that are legal, like alcohol and tobacco. And nicotine and tobacco form are kind of the worst things you can do.
Yeah,
just about.
You can add, what I do is I go into the class and I say, okay, we're going to talk about caffeine today, and I put up the statistics showing like 80% of adults in the U.S. are regular caffeine users, and of those, 80% are essentially addicted to caffeine. I said, you want to know if you're addicted to caffeine or not? How many of you, when you wake up in the morning, if you don't have your cup of coffee, get irritable and grumpy or have a headache? And like more than half.
Every caffeine user, just about. Yeah.
So you are dependent upon caffeine to get you back to that normal state. And one of the things that Roland had told me is like these people, to properly use caffeine, you have to not use it more than about twice a week to get to where it does have that cognitive enhancing function. Because once you are dependent on caffeine, it's like the old adage with heroin. You just need that to feel normal again.
Right,
and
right. Well,
Roland had said to me was, what these people don't realize is if they weren't dependent on caffeine, they wouldn't need their caffeine in the morning to feel like they needed their caffeine.
Yeah, I gave up caffeine, let's say in October of last year, and I've actually given up all stimulants, no stimulant medication, and no chocolate, which has been a hard thing to get rid of. Because I used to drink ceremonial cacao, and I loved it. The pharmacology of it's so cool, and like a lot of serotonin precursors. And I noticed after I drank it every day, when I stopped drinking it, I kind of a little lower mood for a little while, but. I just sleep so much better. I was having profound sleep disruption for years, and just my body's very sensitive. But I've also noticed I have much more energy. I'm much more alert. It's much easier to focus. It's just all the nootropic effects of caffeine were basically, I was just in such a deficit all the time.
Oh, yeah.
Yeah.
It takes about 6 weeks. I asked Roland, okay, if somebody does give up caffeine, what can I tell my students? Like, how long will they have to basically be abstinent to be back to normal, where it would have these nootropic effects if used properly? And he said about 6 weeks. So when I tell that to the class, I say, and if you want to get off caffeine to have it functional for you again, it's about 6 weeks. And they all go, oh, no, I can't do that.
Too long? Yeah, if you're in medical school, there's no way you can just go 6 weeks without being able to think properly. It's just better off staying addicted to it. What you were saying about like getting the heroin, you got to get back to your baseline. You're just trying to get back to normal. I mean, in principle, isn't that how all mind-altering drugs work? I mean, if you take them habitually, is there any? I mean, I would think that's an issue that I've seen with. I've been on a lot of psychotropic drugs, probably 50, and all I really observed was that within a few months I just would end up back to baseline, and then I'd have to go through the withdrawal process when I would come off of them, and it seems to just be an inherent feature of the brain that it's an adaptation machine. I mean, are there any drugs for which like long-term use where you would not expect habituation like that?
The habituation is due to there's this process called desensitization and downregulation. So when your brain and your neurotransmitters are exposed constantly to a ligand, in compensation to that constant.
A ligand is a drug.
A drug.
A ligand is something that binds
your brain will reduce the amount of receptor for that drug. So then you have to take more to have that same effect. And then eventually you desensitize the system so much that it might lose its effect completely. So
that's happened to me with cannabis.
Yeah, most
you just couldn't get high anymore.
Yeah. Like for LSD, if you take LSD on day one, you have a big trip. Take it the second day, you have a moderate trip. You take it the third day, you won't trip at all. It's very fast with LSD if you take it like on a daily basis. But there are drugs that have some effects that do habituate like that, and others where the habituation is on a much different timeline. So like example for an opiate drug, like Oxycontin or something like that. The drug, if you overdose on that, there are a side effect pathway and a therapeutic pathway mediated by two different enzymes downstream of that receptor once it's activated. One of those pathways produces respiratory depression, so you stop breathing. The other pathway is that analgesia pathway. So when somebody first starts taking an opiate drug, they're at high risk of developing that respiratory depression. But then if pain goes worse, the longer they're taking that, they habituate to the respiratory depression effects. And if they're an addict, the same happens. And so
but not the analgesia?
Not the analgesic effect.
That's interesting. Okay.
If they then go off of the drug and they reset and they take the same amount of opiate that they had took either as a recreational abuser or as a prescription user, they become resensitized to the respiratory depression, and then they're at risk of dying from the respiratory depression. So this
is classic overdose. I mean, I've been around the treatment world and addiction for several years, and it's the biggest thing with people that are opioid addicts coming right out of treatment. They're really vulnerable because they just, I've even I was in a treatment center where they just said, look, like if you relapse, don't take what you were taking before you got here. Like that, and that's just basic harm reduction is we don't want you to do it, but if you end up doing it, you're like, this is how people die. And I think there's kind of a common understanding of that. It's like you become resensitized. That's it's interesting that it's separate from the analgesic effects though.
So there are efforts now to make a drug that has engineered out the ability to activate that respiratory depression aspect, but to maintain the analgesic effect. So there's been a few attempts at that.
I'd like to ask about, you were talking about the LSD and then there's the rapid short-term tolerance. I mean, it's massive. You take it one day and then the next day, you're right, like you need 5 times more or something. And then the third day, nothing will happen. I've heard that many people make the claim that psychedelics are anti-addictive. And that was just what came to my mind when you mentioned that, because it is one thing where it's just like it's very hard to habitually use that drug, but I know that all psychedelics don't work that way with that kind of rapid short-term tolerance. I'm just curious if is there a definition of what anti-addictive would be, and to what degree does that apply to psychedelics?
Yeah, it's the mechanism for that really is not really known. In terms of substance use and addiction from a scientific standpoint, we try to model those in an animal that, say you put a rat in a box and there's a lever in the box, and every time it pushes that lever, it gets an injection of a drug. If you put heroin, cocaine, alcohol, methamphetamine, something like that in there, the rat will continue to increase pressing of that drug until it essentially passes out. So that's called self-administration. And so drugs that animals will self-administer are almost always what become addictive in humans. But if you do that with a psychedelic, rats or mice won't do that. They'll give, it's not something that, even if a drug produces unpleasant effects, they'll continue to do this if it has other rewards.
Like cocaine or methamphetamine, or I'm sure very unpleasant in a certain degree, but they'll keep doing more.
Yeah. And with psychedelics, classic 5-HT2A agonists, they won't do that. And they won't do that with MDMA either. So there's no self-administration with psychedelics or with entactogens, which sort of puts them in a separate class, which is kind of mirrored in the human use. Like you give somebody a high dose of LSD for therapeutic purposes. It's like, are you glad you did this? Yes. Would you want to do it again? No.
It's like giving birth. I mean, I wouldn't know from experience, but I mean, it's, I've had that experience where it's like the last thing on your mind is, let's do that again right now.
Right.
Like, I mean, you're just kind of, processing it, and it's like,
so there's very little what I would call addictive behavior associated with psychedelics. And are there people who routinely use it recreational? Yes. But that's not compulsive addictive behavior like you would see with an opiate or with cocaine. It's that compulsion. So on one hand, LSD and psychedelics, they're not addictive. They don't produce compulsive taking behaviors. But then their anti-addictive properties are that sometimes in these clinical trials for substance use disorder, like my colleague Peter Hendricks, who's up at UAB, University of Alabama, Birmingham, he just published on his study of psilocybin for essentially homeless crack addicts. There are people who have been lifelong crack users. They come in, they do their psilocybin session and never touch it again. And
one time and they're off crack. Yeah. And
it could be a combination.
Do we know like a relative idea of the proportion of those people? I mean, is it a substantial, statistically significant number of people, or is it just kind of a noteworthy anecdote?
I think he had, at like 6 months, 12 months, I think there was like a 60 to 70%.
Jesus.
And early on, like there were a percentage that just never touched it again. There were some that maybe did it once or twice, never did it again. And going into that study, the thing was like all of the other studies that had been done at Hopkins and NYU, those were really cherry-picked individuals to maximize success from upper middle class families who had support systems and counselors. And was it really going to work in the real world? And these people, after their therapy, were going back to the streets, their lack of support systems. And so would it work? And a number of these people, they reconnected with families, they sought out social services, they got jobs. So it really showed that in the real world, it can have efficacy.
Yeah, and so like the anti-addictive is not only are you not addicted to this drug, it can cancel out another addiction, which is the holy grail of addiction medicine. I mean, I know there's a lot been made about ibogaine. With opiate, there's a specific opioid pathway there, but psilocybin and crack, as far as I know, don't have much of a connection.
And with ibogaine, nobody's really done the studies with ibogaine and PTSD. There's no reason to think that ibogaine is any better than, say, psilocybin at reducing opiate dependence in veterans with PTSD. Those are studies that need to be performed.
Well, now I know that there's change in drug policy in the last couple months, actually, with regard to ibogaine and bringing that in under the Trump administration. So hopefully, I mean, one of the biggest tragedies of the whole war on drugs is really, in a lot of ways, was a war on science.
Yeah.
And there's all these amazing things that these compounds could do, potentially. Most of it is the stuff of urban legend at this point, but they've kind of escaped the lab again. And science is, like relative to what you might get from an SSRI, where there's billions of dollars of research and all this vetting and all this stuff, where like we have these claims that are vetted, people are expecting the same level of certainty with regard to compounds like psilocybin and ibogaine, and it's just this weird little corner of science that's 50 years behind everything else.
Yeah. It really, there was a lot of research done preclinically. A lot of people assumed when the Controlled Scheduled Act and the FDA regulation changes in 1970 that there was no research on psychedelics. That just halted clinical research. So there were no more clinical studies done to looking at efficacy for treating depression, alcoholism, things like that.
And by clinical, you mean on humans for a specific medical problem?
Right. Yeah. There were hundreds, if not thousands, of studies in the 50s and 60s. So those went by the wayside, but there were a core number of investigators throughout the 1970s, the 1980s, 90s, like my postdoctoral mentor, my father, who really continued to study 5-HT2A receptors, receptor signaling. Like the study of LSD acting at 5-HT2 receptors was instrumental in discovering GQ coupled pathways by my postdoctoral mentors, like the basic fundamental pharmacology from these receptors. And a lot was really known preclinically about what they do in animals, the doses that they give, the types of behaviors that they elicit in mice, in rats, in cats. So there was a lot that was known preclinically, which fed into why when the clinical trials resumed with psilocybin and LSD, the FDA kind of gave a pass on the safety, because we had all the historical data from the 50s and 60s, but then we also had this huge body of research from animals from the 70s and 80s and the 90s feeding into what we knew about the toxicity of these compounds.
Yeah, so like in general, one of the first things you mentioned is when people ask you about psychedelics, they'll say, "Are they safe?" I mean, what can you tell us about this science of the safety of psychedelics?
Well,
or first, maybe we start with, as a psychopharmacologist, how do you discuss safety when it comes to a drug? Like, what are some of the metrics that you would look at?
Yeah, so safety encompasses a rather large area. Safety refers to physical safety, mental safety, cellular safety. Like, is a drug going to kill a cell? So there are tests that you would do first in the drug development pipeline where you have cells in a petri dish and you just add increasing amount of drug until those cells start to die. And then you can say, what's the concentration of that cell or the drug in that cell that starts to die, and how relevant is that to what the concentration in a person would be? And if you have to put a lot of drug in there, you can say, well, we have to put 1,000 times more drug in these cells to get toxicity that we would a human. So it should be fairly safe. So then the next phase is you go to a whole animal and you start with low doses and you go up to very high doses to determine really what it's called adverse events. What level are you going to start seeing adverse events, which could be, say, cardiovascular issues, could be seizures. It's not always death, but like what's the level what you're getting an adverse event? So you'd start off with low doses and you could push it and push it and push it till you say, all right, at 50 milligrams or 100 milligrams per kilogram of this drug, we're starting to get seizures in the animal. But if the translated human dose would be the equivalent of, say, 0.5 milligram per kilogram in that animal, you say, oh, you have a safety margin of 200x before you start hitting any of these adverse events. And then there are also like mental tests that you can do. There are a series of cognitive tests, activity tests that you can give increasing doses of drugs to an animal to see at what point does it now start to interfere with its ability to remember things or to find its way through a maze, and look to see how that compares to the theoretical translational dose. So once you go through all these safety tests and toxicology tests to show that it's not toxic, if you pass all of those, then that's the information you give to the FDA asking permission to be able to give that drug to a human. And then the FDA will evaluate all that data and say, okay, we believe that this is going to be safe for a human to do a phase 1 clinical trial where you now show it's safe in a human and doesn't produce any of these effects.
And that's the purpose of phase 1 is just purely safety in people, right? You're not even necessarily worried, does it work?
Right.
It's just, is it going to kill people, or what are the adverse effects and their incidents and things like that?
Right.
Okay.
Right. And if there are red flags that pop up, like, so many drugs will say, it kills cancer in our mice, and now we put it in a petri dish at cellular cancer. Now it's killing normal cells at a therapeutic dose. Or you put it in a whole animal, and you find out at that point that drug is also activating another receptor that you had no clue was activating, which is causing it to stop breathing. So most drugs will die in that phase of development where they're having these off-target toxicity events.
Right. So, and what you were saying is with psychedelics, we have so much preclinical, which is in animals, research for the last 50 years that they were.
There've been so much by so many people that we knew what the toxic doses were in an animal. We knew what the toxicity in humans were. So the FDA, I don't want to say a free pass, but they let the trials proceed, but there was careful monitoring for any toxic symptoms. And to date, to my knowledge, there have been no real physical safety effects in any of the clinical trials. The one safety issue is the safety of mental condition. And that gets to the regulations in the clinical trials that people who have a relative with first-degree psychosis, probably you want to exclude them because there is an inherent risk of a psychosis-like phenotype resulting, whether it's somebody's first trip or their 100th trip. And it seems to be a constant, like 0.5% regardless. So there is an inherent risk. But the people,
it seems to be 0.5% only in people who have a relative with psychosis, or is that in everybody?
So on everybody who takes a psychedelic, there's about a 0.5 risk of them developing these persistent symptoms afterwards. And when looking at that subpopulation, I would say it's highly enriched for individuals who have first-degree relatives with something like psychosis or bipolar.
I see. So if you took, the 0.5% of all people who have had any kind of persistent psychosis, if you were to look at those people specifically, you're going to have a really high incidence of people with a family history of bipolar disorder or schizophrenia.
Right. So there is always some risk that somebody will mentally go crazy. Right. And that there are some examples of that, but from the clinical trials that have been well controlled, that really hasn't been an issue.
Also, that seems like just from what I know of it from like popular culture, is there are some places and situations that are really not conducive to taking a psychedelic, like at a concert or someone, I talked to someone yesterday who told me, because I told him I was talking to you today, and he's like, yeah, man, I used to take it in high school. And I was like, what? This is the last place in the world I would want to take a psychedelic drug. Yeah. I guess it would make it more interesting, but and so there's a lot of those kind of things where you could have a panic attack. Like you could be traumatized in those situations. And of course, in a clinical setting, one of the benefits of it is that it's more controlled. The environment is more controlled to eliminate something that could be terrifying.
Yeah, I think setting is absolutely critical. That if you're taking a medication in a bad place, you shouldn't be taking it. It's just going to precipitate a bad trip. And for therapeutic purposes, you have to really have like a warm, nurturing, a comfortable environment to have a positive trip. And I think that kind of goes to, today they try to maximize the comfort and the connectedness with the therapist, so you feel you're ready for this in the therapeutic sessions. But MK Ultra back in the day were just taking people in off the street and dosing them up.
This was the CIA experiments with LSD. Yeah,
secret.
Yeah, and what were they doing it for?
They were trying to develop psychedelics as a mind control agent, and one of the main leaders of that was at Tulane University, and he was the chair of psychiatry at Tulane. And apparently, they still have one of the bunkers they used to dose people up back in the 50s in City Park here.
At Tulane and City Park, I believe it.
And I was several years ago, this person who shall remain unnamed, but he was in his 90s at that point, was involved in these early studies with LSD back in the early 1950s, and he would talk about how they would take Amish people who would come into the big city and just bring them in off the street, put them into a dark room with no lights, and dose them up with like 500 micrograms of acid and just watch them.
What? That's horrible. That shouldn't be laughing. But the fact that they're Amish, the fact that they're Amish is a particularly weird detail. Like these are the things when I heard these things in high school and in college. I mean, I would be like, that's preposterous. And like, it
was like, can you break somebody and get them to believe things that aren't true? And this whole MK Ultra program, which
that makes sense. So you get someone who's a true believer and see, can you really change their beliefs? Right. So that's why Amish people.
Exactly.
Okay. Exactly. Yeah. You get religious fundamentalists and okay. Yeah. And so anyway, so that's MK Ultra. Yeah. It's evil that the CIA did it, and they try to destroy all the records.
You're in a horrible setting like this, and you can't help but have a bad trip.
I've heard also there's one like kind of famous. They did a lot of this in San Francisco, obviously.
Yeah.
And they would take people in and bring them into a brothel and dose them, and then I think they would lead them to believe they had murdered the girl, and then try to use that as a blackmail technique. And it's just like, so that's horrifying. I mean, that's just evil to do that to someone. I mean, to dose someone with a mind-altering drug without their knowledge or consent is just evil.
Yeah.
But yeah, so that you were just saying, like, I just had to double click on, because there's probably some people watching this that would not know what MK Ultra was. It sounds like a completely insane urban legend, but we actually have documented evidence from the government records that this really happened. Yeah, in the 60s. So yeah, I mean, that obviously probably would produce a lasting mental effect, like a traumatizing effect, because I assumed, because you're just, these medicines raise plasticity, right? Like to an extreme degree. And so plasticity is neither good nor bad. Like it's just, what is it that you're going to be very impressionable, basically.
Right.
So what's the impression that's going to happen?
Yeah. So my research, we published on it in 2020, and other researchers like Gul Dolan have published on it as well, that there's this window of about 2 to 3 weeks after a single dose of psychedelic, at least in our animal models, where the brain is plastic to the environment that it's in. And during that time, if you put it in a stressful environment, it shapes kind of you lose that antidepressant effect. So
the 2 or 3 weeks after are just as important, arguably, as what you're doing during the acute effects.
So if you take this person after 2 weeks, you give them the therapy, they're able to deal with things. It has this positive therapeutic potential. So that's the integrative therapy. But then if you take the people during those 2 weeks and you put them in this mind control environment and try to control their mind, then you can reinforce whatever this paranoia and stuff. So you could either have lasting therapeutic effects or this lasting PTSD. The plasticity can be for good or bad depending upon what that environment is that you're putting them in.
So I want to put a pin for a second in the role of therapy with these, because I want to come back and ask you about MDMA trials and the denial of that and what you think of that. But you mentioned that like this 2 or 3 week period, and I know about Professor Dolan's research just on like critical periods of plasticity. If I can recall correctly, some of her research pertains to the length of the period that opens as it pertains to the psychoactive effects of the drugs. Could you summarize what we know about that?
So her research converges a lot with our research that shows that there's a particular length of time after the drug is administered where the brain remains in this plastic state, that you can manipulate the environment within the first 2 weeks and essentially persistently affect the animal's behavior after that. But if you manipulate the environment after those 2 weeks, there's no lasting effect. So there's this receptivity to plasticity based on the environment in the first 2 weeks after giving a psychedelic. And the types of experiments that she did were a little bit different than the types of experiment that we did, but they're absolutely congruent in identifying that about that 2 week window of availability for that brain to remain in this plastic state.
So I'm curious about the 2 week period and whether or not the length of that period is dose dependent or substance dependent.
It might be. I think that there is some data that the longer that you're high, maybe the longer it is, but we haven't really seen that. We've looked at the length of the experience in the animal, whether the route that we give it can govern how long that animal is subjectively high. And if we give it in a route where it's subjectively high for, say, 10 minutes by giving an IV, or we give it IP or oral where it's subjectively high for 2 hours, we don't really see any difference in the long-lasting antidepressant effects. So
interesting. When you control for dose,
right,
to the degree that you can do that, yeah.
Right. So there's still, it's an open question. Is the time that you spend high, or that's beyond the side point, is the subjective experience necessary for therapeutic effects or not? But
we'll come back to that.
For argument's sake here, is the time that you are high, is that really necessary or relevant for the persistence of the therapeutic effects? And the data, I think, is piling up that really suggests not necessarily. We've got the preclinical data that show just getting the animal to that point produces these long-lasting therapeutics. The drugs like 5-MeO-DMT that are being used in clinical trials, or DMT, or the IV psilocin, they all will have a very short duration of action, but in their phase 2 trials have all shown an enduring antidepressant effect. And so
of approximately, we think maybe the same
at least 6 weeks, if not longer. People haven't really gone out to a year more in any of these trials. Yeah. So
it's just because of the expense of doing that. And I guess also there's a lot more variables that can add up.
Right. There's really only one company that's developing a long subjective trip, and that's Definium. And they renamed themselves from MindMed a few months ago. And they're developing a proprietary formulation of LSD for generalized anxiety. And so their method, they did a dose response of like 50, 100, and 200 micrograms of LSD. And if I'm remembering correctly, found that 100 micrograms of LSD acutely was the most effective dose to producing these long-lasting therapeutic effects against generalized anxiety. So they're really the only commercial.
That's interesting, because
I'm aware of it.
LSD to me seems to be one of the most anxiety provoking medicines, and maybe it would be interesting to know if there's maybe a resilience that's being built to that.
There's a story behind that involving my father on the anxiety.
Let's hear it.
So some people can take LSD and they don't have any anxiety. It's fun. It's great. Other people, they take it. The first few hours, it's great, but then they get all paranoid and anxious, and it becomes very unpleasant. And my father had noticed this in his rats 30 years ago. The ones that were given LSD repeatedly developed these real paranoid, aggressive behaviors. So he formulated this hypothesis that there was a metabolite of LSD that was activating dopamine receptors, so that after the first part of the trip was over, then it transitioned over into this more dark, psychotic-like, due to this overactivation of dopamine receptors. So he did a lot of experiments and showed that it was the dopamine D4 receptor that was reproducing those effects. He could train rats to distinguish whether they were on the early side of the trip or the later side of the trip. And the later side of the trip, if he blocked dopamine D4 receptors, he could block that. So just last year, we in collaboration were able to make that metabolite that he hypothesized decades ago. And sure enough, it's a very potent D4 agonist. So he thinks, and we've got a manuscript that's been submitted now on the basis of the anxiety and the bad trips of LSD for some people but not others, is that some people produce this metabolite which super activates dopamine receptors and some people don't, which is why some people like LSD a lot more than some people don't.
Yeah, and presumably there's, at the other end of that research, what might lead to some sort of way of inhibiting that or doing something, blocking it in people that to where it could have therapeutic potential. You know, one thing that you've alluded to a couple of times here, and I'd love to ask you about, in my opinion, we're kind of getting into the really juicy stuff, and we're only an hour and a half in, is microdosing. Like that's a common practice. I mean, and there seems to be very little, if any, research, but I think you've done some of it, right? What do we know about microdosing, and what is it, and how is it? What's the protocol, and then what do we know about its effects?
Yeah, microdosing, it can mean different things to different people, and one definition is a sub-behavioral dose taken repeatedly over time. Another definition is 1/20th of a behavioral dose taken over time. There's what's called the James Fadiman protocol, which is, I think, taking 1/20th of a dose every third or fourth day. There's a lot out there. There's people who are doing microdosing with psilocybin, with LSD, and there's very little evidence supporting the claimed benefits. The claimed benefits are enhanced creativity, higher energy, a better sense of well-being. So I was involved in a study several years ago where we had given a repeated low dose of LSD over 2 weeks, every third day or every fourth day, at 5, 10, and 20 micrograms. These are naive individuals. And then did all...
Meaning they've never taken it before?
Any psychedelic before.
Yeah. And so they have no expectation?
No expectation.
What it's going to be.
It's placebo-controlled, double-blind, and cognitive testing was done across several domains, looking at memory, attention, proprioceptive, physical attributes. At the end, none of them could guess if they were on a drug or placebo or what dose of drug, and there were no effects on any of the cognitive measures or real physical measures. So there was nothing there. And the purpose of this actually was not to debunk microdosing. The purpose of this study was to show that low repeated doses of LSD were safe and appropriate for a population of individuals as a safety trial in preparation for going into Alzheimer's patients, and maybe treating MCI with LSD. And so we needed to demonstrate safety.
MCI is mild cognitive impairment.
Right. So this demonstrated safety, but on the flip side, demonstrated that there were no effects.
And so if I'm doing the math right, I mean, you're saying about 5%, that'd be like 5 micrograms of LSD?
5 micrograms, 10 micrograms, and 20 micrograms. We had 3.
So that's a big range. So yeah, and you're seeing the same thing even at 20 micrograms.
Right.
No. And you're measuring cognition. I'm wondering, does that also include mood? Or things like measures of social, so their social life, their sense of connectedness, spirituality, things like that.
Maybe it had like basic mood in the question.
Okay.
But then
so they're not coming in being like, I found God.
No, not at all.
Right. There's nothing, no major change in their life.
And then there have been some studies by Harriet DeWitt and her group at the University of Chicago where she was doing functional imaging and fMRI with 13 micrograms of LSD per person. And they were able to demonstrate that there were changes in connectivity that were visible with the fMRI. So at those doses, there are changes in the brain, but if they're really doing anything, we don't know.
We wanted to translate to conscious experience is the idea.
Right.
So if it's not. Yeah.
And a lot of it, there's a term called expectational bias, which is like placebo effect. And with antidepressants, there's about a 30% placebo effect. If you give somebody a placebo, you tell them it's antidepressant, and then you test them on their depression scores a few weeks later, 30% of people given the placebo will say that the medicine is working. So there's a very powerful placebo effect with psychoactive drugs like this. And so if you have these people microdosing who their friends told them, oh, it works great, take it. I think there's a huge expectancy bias on
the bias is the cognitive element of the placebo effect is that we have this bias that we expect something to go a certain way, and so it kind of does.
So that's my take on microdosing with LSD. With psilocybin, I have now a somewhat different take, because unlike LSD, psilocybin is a pretty good anti-inflammatory, whereas LSD is really not a good anti-inflammatory. And the anti-inflammatory capabilities of LSD are such that at the levels used in microdosing, they're having real effects in our animal models. And if any kind of there's neuroinflammation that's doing anything, or inflammation in the body, the amounts of psilocybin that people are using may conceivably be having an anti-inflammatory effect at those levels, which could be improving the overall body or mental function.
All right. And so you're seeing that in your research now. There's just an effect, and you're, I guess, hypothesizing that it would be related to inflammation, or
So
interesting.
We're nearly ready to submit a manuscript where we did a study using daily sub-behavioral doses of a few different psychedelics to treat rheumatoid arthritis. And each of these included psilocybin in there, had differing effects, but they all had positive therapeutic effects in this model of rheumatoid arthritis at daily sub-behavioral doses.
So, did you also assess for mood alongside while you're assessing for things like that?
No. Yeah.
I imagine if someone's rheumatoid arthritis improves, their mood's going to get better.
Right. Exactly.
And so that kind of creates an interesting conundrum of like, are those things separable? Does it matter? Do we really care? But I mean, I do. I'm kind of curious of like, if it's also having this other benefit, if it reduces neuroinflammation, maybe their mood would get better independently.
Right. So say somebody's got arthritis, they're microdosing psilocybin, which I do not advocate as a medical professional. But if for whatever reason their arthritis improves, their mood is going to improve, the neuroinflammation will go down. So there might be something there, and in clinical trials, that might be something to consider for a future clinical trial, looking at inflammatory biomarkers. And a lot of clinical trials now, in consultation with me or others, they've been incorporating blood draws and other markers to look at biomarkers for inflammation and if those are getting better or not.
That's amazing. Do we know the mechanism for how it reduces inflammation? Is it related to the serotonin pathway, or is it separate? Or
it's through the 5-HT2A pathway. If we use a genetic knockout for the receptor, we don't have anti-inflammatory effects. We block the receptor. We block the effects of psychedelics. So we know it's through the receptor, but the different drugs are doing different things. Like in our models, DMT is not anti-inflammatory at all. LSD, very weak. But psilocybin and psilocin are very anti-inflammatory. And in other molecules that we've looked at, the non-classic psychedelics, they're like some are treating some diseases and inflammatory pathways different than others. So we're in the process of saying, okay, they all have some kind of protective anti-inflammatory effect, but which one works best for asthma? Which one works best for arthritis? Which one works best for inflammatory bowel disease? And so that gets back to how changing a given molecule on a psychedelic will cause that receptor to turn on or off other pathways. And with some psychedelics, there's, I think, multiple anti-inflammatory pathways that are being activated, and some will activate some better than others. And so we're in the stage of sort of figuring that out now.
That's so exciting. I mean, you talk about, we began talking about just like the unintended consequences of basic research. And I know what you're doing right now, you're describing is not basic, but to discover, wait a minute, like we might be able to treat Crohn's disease with this, like, and as opposed to depression, which, I'm excited, anything involving the mind, any kind of new drug that would help people with mood or addiction or anything of mental health is exciting. But I also live with chronic inflammation. And it's just a problem I have all the time. And I just know the mood altering effects of it. I get anxious and irritable all the time, and skin things and whatever. And it's just this kind of basic process that if I can control it with an ice bath or a infrared sauna or an anti-inflammatory diet, it makes a massive difference just in my overall quality of life and my general sense of vitality. And so it's exciting as someone that has this, not that I'm going to go out and start microdosing, because I wouldn't. I've tried it, and you might be interested to know I've tried microdosing psilocybin, and it makes me reliably really irritable. And it's confounding, and it's been consistent across different strains over years. It's been a while since I've tried, because it's like, why be in a terrible mood? It's just really giving you the opposite of what it was supposed to be. Yeah. So I know there's tons more research, but you might find something like a real basic mechanism, and we might lead to lots of like relief for lots of people that are just kind of ailing.
Oh yeah,
from these inflammatory diseases.
It's like 60 to 70% of people are dealing with some kind of inflammatory disorder. Like I myself have degenerative spinal disc disease, and I'm in chronic neck and back pain. There's other people with arthritis. There's people living with eczema. There are so many people with these inflammatory diseases where the drugs that we have, we've got like ibuprofen, which has its...
Or you could take an immune suppressant.
Which then you get tuberculosis. Or
right. It's like, I don't want to get pneumonia.
Skin thins or not really good. And the ones that are becoming popular now that you see on TV commercials are for these biologics, these antibodies against, IL-23 or IL-33. You have to inject those in the clinic, and those are $100,000 a year. They're really not accessible. Yeah. So what's really lacking is a truly disease-modifying, safe, small molecule that somebody can take that is going to treat their inflammation. And so far, we see evidence in a model of cardiovascular, of inflammatory bowel. We've looked at rheumatoid arthritis, asthma. I think it's really promising. And at doses far, far below, like 30 to 50 times below a behavioral dose.
So, for one, thank you for doing this research. Like it gets me excited just to talk about it. But science in general just gets me excited, and the thing that how little information we need to make these massive breakthroughs. But we're kind of been dancing around this subject, and you raised the question earlier, is whether or not the psychoactive effects are important to like the lasting mood effects. And what you're describing here, a 30th of a behavioral dose. Now, again, like maybe I'm sure that people are going to be in a better mood if they're in less pain, But maybe there's something else that's going on more directly. What are your thoughts on whether or not the psychoactive effects are important for like these crack addicts in Birmingham, for example? I mean, I've heard about things like the main variable seems to be the dissolution of the ego, or the oceanic boundlessness and things like that. I mean, to what degree do you think that these things are separable?
I think that there are going to be populations where the subjective trip is going to be necessary, and those where maybe not necessary. And I think most of them it's going to be necessary, because I think if you look at a lot of like the veterans with PTSD, you look at the crack users, you look at people with major depression and other comorbid disorders, there's a lot of trauma associated with that, a lot of childhood trauma or trauma as an adult that has produced this dysregulation of their HPA axis. They're just in this rut that they can't get out of, and the SSRIs and whatnot aren't really very effective. So the therapies like MDMA, like psilocybin, the subjective experience allows them to really confront, face, absorb that trauma, that history, see it from a different angle. That then when the subjective trip is over, the integrative therapy is crucial. Like this is what I was dealing with. I saw my deceased grandmother, and she was so disappointed in me for where I am now in my substance use. And then you explore that in the therapy, and you realize, like, oh my gosh, I'm not alone. I talk to my mother, my grandmother. I have a family. I have a community. They reconnect to that. So having that subjective experience for somebody who has become disconnected, who has had some kind of trauma that they have avoided facing or are unable to just because it's too traumatic to face, they're able to do that. If somebody doesn't have that subjective trip and have that kind of catalyst to recovery, maybe intense psychotherapy within those 2 weeks can get at some of those things. But I think that subjective experience is really the catalyst to the recovery. And if you have somebody whose depression, this is a minor fraction of them though, who are, it's just idiopathic, that they're happy, normal people, normal childhood, there's no trauma, but their brain neurochemistry is a little out of whack, and when they get depressed, they just can't come out of it. And that's a smaller percentage. That is the population that might benefit from the non-psychedelic psychedelics, where you do have that increase in plasticity in both. But in the one case, it doesn't matter if you're having the subjective experience or not, because you don't have any, you don't have these life events. But it might be enough to renormalize some of these circuits that could be deficient, because you have a undiagnosed tumor on one of your glands, and that's producing a synaptic deficit in your brain, and so now you take care of that and re-enhance that. So I think there's a role for both, but it's not really been explored yet in clinical trials.
Yeah, that's interesting. You mentioned like these people that have this idiopathic depression, and they're kind of stuck in it. And I had a really, really long battle with depression and with trauma, and I've been fairly public about it on this podcast. It's kind of how I've become interested in the mind. And I can imagine the thing is, what you're saying, I was thinking two things. One was, well, yeah, I mean, if these people could technically benefit without the subjective effects, they probably still arguably benefit more with them.
Yeah.
Like there doesn't seem to be any reason to exclude that. But also, you still need to make sense of the depression. You still need to make sense of what you've lost, right, and of the shame that comes from people not getting it and thinking that you're just not working hard enough or whatever, and of the life you've lost, of the experiences you've lost, the relationships you've lost. I mean, it becomes, at a certain point, it becomes its own trauma, and you have to make sense with that or come to peace with it and come to love it or whatever. I mean, it's a kind of weird thing where these compounds can facilitate connections where it's like you can anthropomorphize a disease like depression and then come to love it and give it a hug. Or things like that, where it's just there's no way to really, I want to ask you about this MDMA trials. That they're their phase 3 trials. I think it was 2 years ago. They got rejected by the FDA for approval. What's the name of the company? Is it Lykos?
Oh, it's now they changed from Lykos to. Oh, there've been so many companies that have changed names recently.
Right.
Oh, I can't remember their name now.
This was the first. It was backed by MAPS, and it was the first real push for approval for MDMA. And I would love to know your opinion on the reasons for the denial of the approval by the FDA. It to me, my layman's assumption or my layman's impression of it is that the FDA doesn't really understand how to standardize therapy. Like that seems to be a real sticking point. That therapy seems to be an indispensable part of how this drug is meant to be used, and because it's a different therapist every time, there's a different human element, and that there was some lack of standardization. But maybe I'm wrong about that.
Yeah, I'm not really certain of that. They did ultimately release the FDA document on the comments on why it was denied their NDA, and I know my personal opinion is, there's a lot of opinions out there and a lot of smoke and mirrors, like with what happened with the phase 2 trial sites up in Canada with some people being taken advantage of. But I don't ultimately think that factored into the FDA's decision. And I think a lot of what factored into the FDA's decision was that certain metrics were not gathered that are required of all psychoactive drugs. For example, the adverse events was a big one. Not all trial sites collected adverse events because I think there was maybe a misunderstanding of what Lykos thought was an adverse event versus what FDA defined as an adverse event. Whereas the FDA defines, in my understanding, an adverse event is anything that's out of the realm of the ordinary. Whether it's subjectively positive or negative for that person, that's an adverse event. Whereas Lykos may have been looking at adverse event as something that's negative that's happening, that they had euphoria, so that euphoria wasn't counted as an adverse event.
In my layperson's understanding of what adverse means, I wouldn't count euphoria as an adverse event. So it's interesting but they would at least need to document that because somehow that's not intended.
Right, and and that events like that are required of all CNS active drugs that the FDA goes through. So it's a bit of he said, she said from Lykos's perspective, and the FDA. Like Lykos say, oh, they moved the goalpost. The FDA says, no, we didn't. It ultimately comes down to, I think it was a new company, had never gone through an approval process. It was a new type of approval for the FDA. And mistakes were made potentially on both sides. And I thought it was interesting that the FDA advisory panel, that the two people that were on it who voted for approval were the external members who were invited who said, but we don't have anything. So it'll be.
What do you mean? What do you mean they meant that they don't have anything?
Therapies to treat PTSD.
Right. Right. There's a sense of urgency that we need to bring this to market. And that's a valid, valuable perspective. I think that also should be counterbalanced with an understanding of safety because if you rush to bring things to the market because we need them, you could kill people.
Right. And I think maybe Lykos was relying on, it's been in people for decades. And then the FDA was like, being in people and getting anecdotal reports is not the same as asking them when they're high on the drug in your therapy session. So I'm not quite sure what's going to happen. I don't think that they are going to do an additional trial. I think they're going to resubmit for that NDA by trying to respond point by point to what that notice was that the FDA issued, and we'll see. Do I think MDMA will ultimately be approved? Yes.
No question.
I think it will. And I think psilocybin, there's a likely possibility that psilocybin will be approved by the end of this year or early next year. That Compass Pathways, I think they've dosed their last patient in their phase 3 trial. And they released their top line data a couple weeks ago on their phase 3. So
what did that data show?
For treatment-resistant depression, it showed pretty impressive enduring effects for psilocybin at, I think it was 12 weeks out. Yeah, and so at the clinical endpoints. So they've done everything.
From a single administration, like a single dose. Yeah, so that's better than taking SSRIs every day.
They've got their phase 3 trials are now complete. So now they're putting together their new drug application package, and if everything continues to go on track for them, I would anticipate it to be submitted to the FDA by the end of the year or early next year. And then if the FDA approves it, then they have to work with the DEA to reschedule.
I think that it seems like there's a lot of political will in the Trump administration to tell the DEA to cooperate, which it historically has not been willing to do.
Yeah.
But it's also generally, like I think for a long time, it was just a massive waste of resources to spend more than half of their budget going after marijuana because they just refused to reschedule it. Yeah. From Schedule 1. Do you anticipate that any other psychedelics, classic or otherwise, might be approved in the next, say, decade?
I think probably, Compass is going for treatment-resistant depression. Usona is not very far behind Compass at all, and they would be anticipated to submit their new drug application next year as well. They're just going for major depression. So those two, Definium is in the midst of their phase 3 for generalized anxiety, and they've got breakthrough status by the FDA. So
that's psilocybin?
That's LSD.
LSD, right. Definium, that's the one you mentioned.
They could be approved, say, in 2028, pending positive phase 3 trials. AbbVie bought Bretasolocin, I think, from Gilgamesh for $1 billion earlier in the year. Eli Lilly just bought Atai Beckley for their nasal DMT, 5-methoxy DMT, for something like $3.8 billion.
That's 5-MeO.
For 5-MeO.
Yeah.
So both of those assets have been through phase 2B, showing efficacy. So what Lilly and AbbVie need to do now is to do a large phase 3 randomized controlled trial at multiple sites, costing hundreds of millions of dollars. So if they get those trials going, say over the next year, those trials should last about 2 years. So you would see within about 3 to 4 years, maybe one of those assets being approved. You have there's a company now called Helus, Helus, which has deuterated.
That's a good name. Helus. That's a good name. Yeah.
Deuterated psilocybin. So it's got like heavy water hydrogens on it.
Deuterium. Yeah.
Has a slightly different pharmacokinetic profile than psilocybin. They are now in phase 3 trials this year. So if they're successful, they could have something approved within the next 2 to 3 years. Trying to think who else is in phase 2B, early phase 3. I think those are the only companies that are in phase 3 now, or would be going into phase 3. There's Reunion, which is developing a prodrug of 4-hydroxy-DIPT for postpartum depression. I think they've got very encouraging phase 2 data. I don't know if they've started their phase 3 yet. So I think within the next 3 to 5 years, we could see several psychedelics come on the market for treatment-resistant depression, major depression, postpartum depression, PTSD,
addiction,
substance use.
Well, the thing is, once something comes on the market for one thing, you can also kind of use it off-label.
Right. Depending if the FDA allows that off-label use.
Interesting.
But
so there's a list of things that are approved for off-label,
Yeah.
but that okay, all right.
So I think there's a lot of development and excitement out there. I think what the main challenge is going to be now is not acceptance or moving to trials, but what's the insurance reimbursable going to be? What's the access going to be? Is insurance going to reimburse a psilocybin therapy, a course of therapy? Are they going to reimburse for an MDMA course of therapy? It might be $30,000 for a psilocybin course of therapy. And are they going to do that when a monthly prescription for Prozac is going to cost them $5?
$8, something like that. Yeah.
Are they going to do that? And then the delivery. There is no system of care delivery. I don't think it's going to be where you'll have a rescheduling across the board. I think you're going to have it's going to maintain Schedule 1 for recreational, but in certain like treatment centers, it'll be descheduled to a 2.
It's like in geographic location.
Right.
That's an interesting way of doing it. Yeah.
A designated treatment center where you're going to have a facility that's going to be certified to be able that. None of that's out there. So we might be in the position of having psilocybin approved, the DEA in the process of descheduling for therapeutic purposes or rescheduling, but there's going to be no infrastructure for that delivery.
Yeah.
Where you're not going to be able to just have a psychiatrist in their office be able to get psilocybin, because you're going to have the safety. We don't know what the REMS is. The REMS is like the after safety program. What are the monitoring of the patients? How long are you going to keep them in the clinic? What kind of clinic are you going to have? Like they have with ketamine, the nasal spray esketamine. You have to stay in the clinic for 2 hours. You can't take yourself home. Somebody that you know has to take you home. You have to have a follow-up appointment. So what are those going to be with the psilocybin therapy? Those haven't been defined yet. So before
and they won't be until the FDA approves it.
Right.
Then that's all to be worked out with the DEA.
Right. So you're not going to see commercial availability until all of those are worked out.
Right.
And that's the day that if the new drug NDA is issued, that's going to be great. But that's again the start of another long process to put all these other things in place. And so it's going to be a while between when the FDA approves it and when it's going to be available, because there's a lot of issues to address.
Do you know of any research on like medicinal benefits of other compounds we haven't mentioned, like mescaline? I think that's one of the ones that hasn't, that I know of just off the top of my head, that hadn't come up.
There are a couple organizations who are studying mescaline. One of the issues with mescaline is its low potency, its propensity to have people vomit, and it's also an issue with preservation of the peyote cacti, which is becoming an endangered species. That as mescaline would become more popular, you get more people going in and raiding these
peyote gardens
lands for the peyote cacti. So that's why in a lot of the legislation in California, there's decriminalization of a lot of substances, but not mescaline, in order to protect the peyote cacti. So it's a little bit,
and just outright in the drugs. I know mescaline's abundant in San Pedro cactus.
Yeah,
it's all over the American Southwest. I mean, it's not limited at all.
But discussions, like in the early 2000s, the very first studies using psychedelics that were done by Roland Griffiths and Charlie Grob back in like 2006, 2009. There was a meeting like, what drug do we use? And it was because the Hefter Research Institute was funding these studies. So we were there. Do we use mescaline? We thought no, because it's going to make people sick and vomit. It's not very potent. So you don't want to give sick cancer patients something that's going to make them feel
vomit-inducing.
So then, is it LSD or psilocybin? LSD is going to last just way too long, and it's got such cultural baggage. Like, you've got your grandmother was cancer, and now you're giving her LSD. So we can't do that. So then at default, it sort of landed on psilocybin. Psilocybin, it's got a 6-hour time course. It's been used by indigenous populations for decades. It's not going to make them sick. And it's easier to justify giving medicinal psilocybin to a cancer patient than it is the others. So that's kind of how psilocybin was chosen as the drug to use in these initial human studies back in the early 2000s. In the studies that were funded by the Hefter Research Institute, which really funded all of Roland's studies, the Hopkins studies, the NYU, up until very recently, and still active funding some new investigator initiatives.
Speaking of the purging, the vomiting, our mutual friend Larissa Carnero, who connected me to you, when my episode with her talking about, we talked a lot about ayahuasca, which contains DMT, but it's a plant. It's taken ceremonially. Is known as a purgative. It will cause purging of vomiting or diarrhea. And she mentioned that there's some people that think that actually might be part of its mood altering effects, the way that affects the bacteria in the gut, that we produce most of our serotonin in our gut. Do you think that there's any there potentially, anything worth studying of like how that, I mean, it can make people profoundly sick, but we do produce a lot of serotonin in our gut, do we not?
Yeah. A lot of the purging is really what's in the intestine. It's not what's in the gut. Point. And the gut produces the serotonin from what are called enterochromaffin cells, and the serotonin is released directly into the enteric nervous system and the blood system. It's not released like directly into the lumen of the intestine. And a lot of what's believed to be one of the main purgative mechanisms of, say, a chemotherapy is this massive release of serotonin from these enterochromaffin cells in the gut that activate serotonin 3 receptors. And serotonin 3 receptors signal to the vomiting center in the brain to become very nauseous and vomit. So it's this
interesting
that
serotonin does everything.
Yeah.
That's incredible. I mean, inflammation, vomiting, mood, learning, memory.
I don't think there's a single physiological behavioral process that you can think of that doesn't involve serotonin at some level.
Wow. Yeah, that's interesting. And so, conceivably, I mean, researching psychedelics could lead to something that would help people on chemotherapy vomit less.
Yes.
Conceivably. I mean, I think it would probably be a fool's errand to go looking for that, maybe, but that's conceivable at that point.
Yeah,
that's really exciting.
It's used to block chemotherapy, nausea, and vomiting on Dancitron. Zofran is a selective serotonin 3 receptor antagonist.
Wow. So I know we need to bring this conversation to a close, regrettably, because I've really enjoyed it. I'd love to. Next time I'm in New Orleans, I got to connect with you. We got to go to Cafe Du Monde or something. But I just wondering, after having this conversation, there's so many things I'm excited about that I've heard. I'd like to know, is there anything that we've discussed or haven't discussed that you're the most excited about coming up just in science or in medicine?
Well, in the field of psychedelics, I'm most excited about the anti-inflammatory aspects of psychedelics, and I've been ringing that bell for over 15 years, trying to get people to become interested that psychedelics are anti-inflammatories, and like approaching colleagues, try it in your TBI models and other inflammatory models, and I was just,
TBI is traumatic brain injury.
Right. It's like, it's interesting, but we're not interested in psychedelics. And now I'm seeing psychedelics being studied for traumatic brain injury and other things. And so people are finally recognizing that psychedelics are really good for things outside the brain and the psyche. And so that's really exciting for me, seeing that the 5-HT2A is a target for really whole health, whole body health, not just to treat mental like depression or PTSD. So I'm excited to see the use of psychedelics going off in those directions.
Remind me, I don't know if you actually told us the story of how did you actually stumble upon the anti-inflammatory effects? What was the experiment that you were doing?
So it was after Hurricane Katrina, and I was looking for a postdoc, and this fellow whose mentor was leaving just showed up in my office one day, wanted to know if I was looking for a postdoc because his mentor was leaving. And he had done research with serotonin 2A receptors and hypertension when he got his MD PhD in China. And I said, sure, I am. And he had experience with serotonin 2A. So I hired him, and we were primarily doing cell-based assays to try to find outcomes so we wouldn't have to keep taking the brains out of animals. And his background was cell models of atherosclerosis and inflammation, which I thought was interesting but had no relevance to psychedelics. And at one point, his name was Beng-Ning Yu. It was he had the idea. He was an expert in this cell-based atherosclerosis assay. He said, I want to do this assay and test the psychedelic in that, see if it's anti-inflammatory. I was like, that's crazy. Psychedelics, the other 2As in the brain and whatnot. So I said, go ahead and do it. And my now ex-wife at the time, she was my wife at the time, she ran the cell culture facility here that routinely made these prohibitively expensive cells that we needed for that. So I got the cells, Beng-Ning set up the experiment where we added the inflammatory agent, added the psychedelic, and super, super potent anti-inflammatory effects. So then I went back, I repeated it, and we knew we had something there. And then what started as kind of a curious experiment ended up now being more than half my research. But if it had not been for Hurricane Katrina, my ex-wife, being the director of this core, and a supplier of one of the reagents we use, being able to supply a reagent we're no longer able to get, we never would have discovered this. So it was a real serendipity, being in the right place in the right time at the convergence of the universe wanted this discovery to be made. So it was. That's
beautiful. When you're saying that, one thing we didn't really talk about is that a lot of people who use psychedelics will report religious experiences or spiritual experiences, and I'm wondering if do you see any divine providence in that, or do you tend to look at the world that way as a scientist, or do you just kind of stick to what you can measure?
I'm a fairly strict reductionist, atheist, but that being said, the universe does work in mysterious ways. So it's quite a coincidence. I'll just leave it at that.
Yeah. I mean, I think it's really a matter of perspective of whether you're willing to be open to say, hey, that's incredible. And just to basically, like you said, like if it hadn't been for these things, this wouldn't have happened. And you can kind of celebrate that on its own without needing to imply agency.
Mm-hmm.
But it is a really remarkable thing. I mean, it's one of the things, like I've said, I love about science is just, if you look at things like archaeology, just the little tiny little things that we find, and we can extrapolate massively consequential things from deduction and reduction. And it's a really cool thing. I've had so much fun nerding out with you about science and just neuroscience in the mind and psychopharmacology. And would love the chance to talk to you again sometime. And there's so much more that I would love to learn. There's so many more basic questions I'd love to learn about psychodynamics and pharmacodynamics and pharmacokinetics and the differences and things like that. But I know you have to go. So thank you very much for your time.
Oh, thank you. You're welcome.
Okay. Thanks, Charles.